All right, coming back at you for the last part of our lab, and you'll see now I have activity C opened up right here on the side because I want you to be able to fill in these Punnett squares as we go. We did practice on this in class, but if you're still struggling, please watch this quick video. I'm going to do the first example with you, okay? So we're looking at Punnett squares to model the different possible genotypes. So the parent genotypes we write across the top and the side, and then in the boxes are where our possible offspring are. So in our example here, we have our two parents, and then instead of 4, they give us 5 possible offspring, but it's the same idea, you guys, just 4 offspring in this one. Okay, first square is filled in for us. So right here, we already have our first one filled in. We can do this just like we did with our codon chart in the last unit. We take our whatever's up on top and we drag it down. So like capital F is going to drag down here. Lowercase s drags over and those combine. So if we complete the first one over here on the next side rather, you're going to have a capital F and another lowercase f. So it'll be, change my color. It'll be capital F and then lowercase, not an F. Okay. Right here we have a capital F and the lowercase f. So it's going to be another capital F, lowercase f. And you have to make sure you're doing these properly, because if you put one capital that's not capital or one lowercase that's not lowercase, you're not going to have the correct genotype. And another capital F, another lowercase f. So that's what we're going to put right here again. Okay, so those are ones we just filled in together. So now that that square is completed, we can look at our offspring up here. So what do we think our parent mouse should have been right here? You should be answering this in your mind right now. What should your parent mouse have been? What should this parent mouse have been? You guys have had some practice with the alleles and genotypes by now. You should kind of have a better understanding. What are the genotypes of the offspring? Well, if we look at our offspring, all 4 offspring are the same. Okay, so we can see all 4 offspring are the same. So their genotype is all FF. Personally, I just like my font to be a different color when I'm answering questions. What percentage of the offspring will have black fur? Well, if our capital allele is present in all of these, we're looking at the pattern of inheritance for dominance. So anytime we see a dominant allele in these cases, we're going to have black fur. So if we see black, oh, this one's black, this one's going to be black, this one's going to be black, they're all going to be black, right? So 100% of them would be black. Of the offspring right here, are any of them going to be white? Okay, make sure you tell us why. So what do you think for that one? Okay, so now we've got that set up. We have a black and a white mouse we need to set up in here. So I'm going to drag my hybrids from the last experiment out. Take a pure white and a pure black, as it says. Okay, that's what our Punnett square is telling us right here. And then we have to click breed several times. So we're going to do this and try and get, I would say, up to 100. It makes it easier. So I'm going to go up until I have 100. Whoa, okay, so now, were your predictions correct? Did we say that 100% will be black? How many are white? Ooh, what did you write down there? So were they correct, yes or no? So you're going to do the same thing, fill in the Punnett square once you have it filled in for these 2 examples, then run the simulation using those 2 examples, and tell me if your predictions were accurate, okay? And then you'll complete this one more time with another set of data, okay? And then don't forget about your CER at the end. Good luck!